Hello and welcome to one of our most important topic of discussion. I'm Dr. Ray, delivering my lecture as from Wise Owl Communications. The first topic of our discussion for today is important recombinant DNA technology enzyme. So, so in order to talk about the recombinant DNA technology enzyme, one of the most important enzyme that we need to talk on is polynucleotide phosphorylase that was discovered from extract of Azotobacter azyl by Grunberg, Manago, and Okawa. Polynucleotide phosphorylase, called PNPase, catalyzes the synthesis of long-chain polynucleotide RNA, which runs from 5' to 3' directions from nucleotide diphosphate as precursors, and reversibly, it catalyzes the phosphorylated cleavage of polyribonucleotide running in the 3' to 5' directions, with the release of orthophosphate in the presence of inorganic phosphate. So one need to understand that whenever there is involvement of phosphorylase, there will be the involvement of ATP. So therefore, PNPS or polynucleotide phosphorylase is basically a bifunctional enzyme, and it actually functions in mRNA processing and degradation inside the cell. Structural and physiological basis of studies of this particular enzyme have actually showed us that it is actually formed of subunits of the arrangement of various subunits that actually varies from species to species, which would actually alter the perspective as a whole. Now, it's very interesting to note that this enzyme has a property to catalyze not only the synthesis of mRNA from mixtures of naturally occurring ribonucleotide diphosphate, but also that of non-naturally occurring polyribonucleotides. Now, as I talked about that, it plays a pivotal role in processing of the mRNAs. So therefore, one has to understand that this entire processing, which runs from 5' to 3' from coding and non-coding strand from 3' to 5' directions, it actually starts off in the both region. We have the promoter, the RNA coding region, and therefore it processes to produce a mature RNA transcript which runs from 5' to 3' directions. Henceforth, these enzymes have the ability to catalyze not only the synthesis from the mixtures of naturally occurring ribonucleotide diphosphate, but also that not naturally occurring polyribonucleotides. So, in our DNA cloning technology, it has been used to synthesize the various radio-labeled polyribonucleotide as a whole when we talk about this nucleoside diphosphate monomers. So even in E. coli, we have actually studied that it is tried to understand the mode of action of this particular enzyme, as it has been mentioned, that actually the regulation depends on the involvement or synthesis of the production of mRNA processing, either by addition of ribonucleotide to the 3' end, or just by cleaving the bases that runs in the 3' to 5' directions. So therefore, the functions of PNPs, that is a polyribonucleotide phosphorylase, it depends upon the inorganic phosphate as a whole, as well as the presence or the concentration. When there's a high concentrations of the PI, then that time what happens? That time, as we can see, that there will be an involvement of higher PI concentration. It initiates the addition of one or more nucleotides to the existing poly A tail, the polyadenylated tail. And in the process, they try to generate this inorganic phosphate. On the other hand, when this dissociation of the PNPS occurs at the 3' end, again, there's abilities of PAF1. So therefore, the interplay of the PAF1 and PNPS and the concentrations of the PI as a whole regulate the maturations of mRNA, as well as, in turn, the degradations of it. So therefore, the next enzyme that really comes into the play is that this when we talk about this of this polynucleotide kinase, we have to get into the understanding the different functions that actually bridge to the localizations or understanding of the polynucleotide phosphorylase as a whole. Now, this polynucleotide phosphorylase, therefore, it is involved in mRNA processing, degradations in bacteria, plants, and also in human. It also synthesizes a long, very highly heteropolymeric tails in vivo, as well as accounts for all of the observed residual polyadenylations in the poly A tail polymerase 1 deficient strains. When we talk about the polynucleotide phosphorylase, it also functions as part of RNA degradosome in E. coli. And RNA degradosome is actually a multi-component enzyme complex that actually includes the RNase E, it's an endoribonuclease, the polynucleotide phosphorylase as a whole. We have also another involvement of RHLB, that is a helicase, which is a dead box helicase. There's a glycolytic enzyme enolase, and this entire complex, it actually helps in catalyzation process of 3' to 5' exonuclease activity in the presence of ATP, and therefore depends on the concentrations of the inorganic phosphates. So in eukaryotes, the exosomes, they are located in somewhat in the nucleus or in cytoplasmic matrix. Now this degradosome or the mRNA degradations is actually occurring in bacteria, and the exosomes in the eukaryotic system, they're all associated with the processing, right? Control and also the productions of mature RNA transcript in turn plays an important role in our DNA technology. So whenever we talk about this recombinant DNA cloning technology, it has been used to synthesize this radio-labeled polyribonucleotide phosphorylase from nucleoside diphosphate to its monomeric form. So another most important kind of enzyme that plays an important role in our DNA technology is the involvement of deoxyribonuclease. Now, shortly we call as a DNase, a nuclease enzyme that catalyzes the hydrolytic cleavage, the liberation of the H2O, and the involvement of water molecules out there, or the phosphodiester bond in the DNA backbone, and they are known to be as a deoxyribonuclease. Now, based on the position and the mode of action, these enzymes, they are broadly being classified as an endoribonucleases or endodeoxyribonucleases, which cleaves the DNA sequentially internally when we utter the word endo. And there's an involvement of exodeoxyribonuclease. So exo means we're talking about the terminal nucleotides. The cleavage is occurring at the terminal site. And obviously, it's a ribonuclease activity with the removal of the hydroxyl radical. So unlike the restriction enzyme being involved, the DNase does not have any specific recognition site. So we all know that now the restriction enzyme, they have the three properties, right? The cleavage at the particular, upon interacting with a particular recognition site, they bind, they cleave, and they modify the system with their generation of certain ends, which are either the blunt end or the cohesive end or the compatible end as a whole. So there is a wide varieties or array of deoxyribonucleases known which have a different substrate specificity, chemical mechanism, and biological functions. So when we talk about that, so basically, unlike that of the restriction site, the cleavage of the DNase, it does not hold of any specific recognition site. But yes, exception rules, there are certain which we will be discussing one by one. So for example, when we talk about the deoxyribonuclease as DNase 1, an endonuclease which actually cleaves a double-stranded DNA or a single-stranded DNA. So the cleavage preferentially occurs adjacent to the pyrimidine C or T residues. So therefore, the major outcome or the product being formed is a 5' phosphorylated bi or tri or tetranucleotide. So therefore, there's an involvement of a cofactor molecule. The divalent ions are there, and they are important tool in proofreading, in processivity. So they are the calcium, manganese, or magnesium for its activity, and therefore creates a certain blunt end or one or two nucleotide overhang sequences. So when we talk about the DNase 1, is the most widely accepted enzyme in the cloning technology to remove the DNA contaminations from the mRNA preparations. So in order to construct certain cDNA library, the northern blot analysis, RT-PCR, all these things requires the involvement of the DNase. So the mode of action of the DNase is not based on the recognition sequence, but rather according to the divalent cations to be used, the processivity are being monitored. Now, let me share with you guys that in the presence of magnesium ions, the DNase 1 hydrolyzes each of the strand of the duplex DNA that produces a single-stranded nicks in the DNA backbone. So what's a nick? Nick is actually the cleavage at different specific sites that generate various random cleavages. So on the other hand, the presence of these manganese divalent cation, we have the DNase 1 cleavage, both strands of a double-stranded DNA, which approximately ranges the same side production of the blunt-ended DNA fragments with one or two overhangs on either side. So the two major DNAs which have been found in the metazoan, if a question comes in relative to the example, it would be DNase 1, the deoxyribonuclease 1, or deoxyribonuclease 2. Well, the most interesting among the facts, let me just share with you, is another, that is phosphatase. Now, the enzyme in modification of the phosphatases and methylases, they also play an important role in the RDNA technology. Now, the word phosphatase, when ASE is attached to any of the enzyme, we call as it's a removal of the substrate. So phosphatase catalyzes the cleavage of the phosphate or the removal of the phosphate group from the substrate by the involvement of the water molecule or by the mediated by the hydrolytic cleavage. So therefore, this reaction is not reversible. It actually shows a totally opposite activity from the enzyme like the kinase and phosphorylase that adds a phosphate moiety to their substrate. So the basis of their activity, there are two types or categories of phosphatases. We have the acid phosphatase and the alkaline phosphatase respectively. So in both the form, the alkaline phosphatase, they are the most common, they are the most special classes of phosphatases that removes the phosphate either from the protein, which is called the phosphoprotein phosphatases. Right. Okay. So among the categories of various phosphatases, we have the acid phosphatases and the alkaline phosphatases. Acid phosphatases, they shows its optimal activity at the pH between 3 to 6, and the lysosomal enzyme and that hydrolyzes the organic phosphate, liberating one or more phosphate group. They're all found in the prosthetic epithelial cells or erythrocytes or prosthetic tissues, spleen, and kidney. So the alkaline phosphatases, the homodimeric enzyme which catalyzes or which caters the reaction like hydrolysis and transphosphorylation of the phosphate monoester. So they actually shows their optimal activity at the pH, which ranges to about 10, right? Okay. Now, coming to the alkaline phosphatases, once again, we get an idea about the first zinc enzyme that we discovered having 3 close space metal ions. The 2 zinc ions and one of the magnesium ions in which the zinc ions are being bridged by certain amino acids like aspartate 51. The mechanism of action is actually based on the reactions where a covalent serine phosphate intermediate is actually being found and actually being produced to inorganic phosphate and an alcohol. So in human body, if you kindly see to it, that actually contains 4 isoforms, in which the 3 are very tissue-specific isoforms. There's a placental, germinal, and intestinal, and one is non-specific isoform. So therefore, the gene that encodes for tissue-specific isoform, they are present in chromosome number 2, while the gene for a non-tissue-specific form, they are localized in chromosome number 1. So during any kind of post-translational modification, the alkaline phosphatase is actually modified by means of N-glycosylation, so which undergoes a kind of modification through which there is uptake of 2 divalent cations called zinc and one of the magnesium ion which occurs and which is actually important in forming the active sites of that enzyme. So therefore, the alkaline phosphatases, they're isolated from various sources like microorganisms, tissues of different organs, connective tissues of invertebrates and vertebrates, and also the human body. Another most important are the ANA group of enzyme, which actually plays an important role in modification, is methylase. Very interesting. So the word methylase or the methyltransferase or methylase actually is involvement of the transfer of the methyl group to its substrate. The process of transfer of the methyl group to its substrate and the epigenetic regulations, the methylation is actually a common phenomenon in the DNA, and also to bring about some non-specific modifications called covalent modifications, the protein structures. So methyltransferase uses a reactive methyl group, and there's a bound to the sulfur moiety as S-adenosyl methionine called SAM, which act as a methyl donor. So methylation normally occurs on the cytosine residue, which is called the CpG island rich region. And in protein, the methylation occurs on the nitrogen atom, either on the N-terminus domain or on the chain of the protein. So DNA methylation regulates the gene expression, that's both the gene silencing or without the change in the DNA sequences. So it's just merely a chemical modifications or epigenetic mechanism of influences or regulations that's been done. So in bacterial system, this methylation plays a very important role in preventing, in shielding their genome from degradations by restriction enzyme, by chopping down, by cleaving. So it's a part of the restriction modification system in bacteria. Coming to the most interesting classifications of methylations or methylase transferase, they can be classified as in three categories. One, the methyl 6A generates an N6 methyl adenosine, N4C generates N4 methyl cytosine, M5C generates N5 methyl cytosine. So M6A and 4C methyltransferases, they're primarily found in prokaryotes, and these enzymes are responsible for methylations of the DNA sequences in order to prevent the host from digesting its own genome via the restriction enzyme. Okay, so when we talk about the mechanism, the mode of action, or the involvement of how it's been done, the restriction enzyme, as for example, the EcoR1, they cleave within the recognition sequences if the DNA is unmethylated. So on methylation by methylases, the restriction enzyme, the EcoR1, is actually inhibited from cleaving within the restriction sites. So some examples of methyltransferases are DNA adenylyl methylase, which is called DAM, or histone methyltransferases, or O-methyltransferases, etc. So DAM methylase is actually used in recombinant DNA technology, which can methylate adenine in the presence of 5-GAT-C3 domain. And this enzyme can methylate a newly synthesized DNA strand on specific sites. Yes. So another to note, one of the interesting categories of DNA enzymes in modification is ligases. So DNA ligase catalyzes the formation of phosphodiester bonds between two deoxynucleotide residues of two DNA strands. So therefore, DNA ligase enzyme requires a free hydroxyl group at the 3' end of one of the chain and the phosphate group on the other. So the E. coli and the other bacterial DNA ligases utilizes the involvement of NAD+ as an energy donor, whereas in the T4 bacteriophages, the T4 DNA ligases uses ATP as a cofactor. So in both the cases, the role of the DNA ligase is somewhat to seal the nicks, the cleavage portions, right, in the backbone of the double-stranded DNA after the lagging strand be formed to join the Okazaki fragment. So the joining process is very vital and pivotal, essential for the normal synthesis of the DNA and for repairment of the damaged DNA. It has been exploited by genetic engineers to join the DNA chains to form the recombinant DNA molecule. Usually, single-stranded breaks are being repaired using the complementary strand as a template, but sometimes double-stranded breaks can also be repaired with the help of the DNA ligase 4. So the most widely accepted DNA ligases, which are being formed from the T4 bacteriophages, is T4 DNA ligases, which needs ATP as the main cofactors. So let's understand the applications or the involvement. So understanding the mechanism of DNA ligases, ATP or NAD plus react with the ligase enzyme in order to form a covalent enzyme. So AMP complex, in which AMP is actually linked to the amino group of a lysine residue in the active site of the enzyme through a formation of the phosphoamide bond. So the AMP moiety activates or triggers the activations of the phosphate group at the 5' termini of the end DNA molecule to be joined. So it's definitely called a donor. And the final step is a nucleophilic attack on the 3' hydroxyl termini on the activated phosphorus atom, which acts as an acceptor molecule. So therefore, a nucleophilic after the accepted donor relationship and the phosphodiester lineage or the linkage is being formed, and therefore the release of the AMP. The reaction is driven by the hydrolysis of the pyrophosphate moiety released during the formation of the enzyme adenylate complex and the involvement of two high energy phosphate bonds, which are actually spent in formation of a phosphodiester bond in the DNA backbone with the ATP serving as the energy source. Adenylate and DNA adenylate, they are very important intermediates for the formation of the phosphodiester bond. Now, when we always look into the application of the DNA ligase, the enzymes are primarily used for sealing purpose, for joining purpose. Following other examples of application of the ligase enzyme, like to construct a recombinant DNA molecule in molecular cloning, joining of the adapters, linkers, for conversion of a blunt to the overhang or cohesive or compatible ends, cloning of a restriction DNA to the vector to construct a recombinant vector. So all these things would be a result of that. Coming to another most interesting modifying enzyme is the polynucleotide kinase. The polynucleotide kinase is basically, as you can see, is a homotetramer with the phosphatase activity at the 3' end, the kinase activity at the 5' end, with a tunnel-like active site. So the active site, which has a side chain which interacts with the NTP donor beta phosphate and the 3' phosphate of the acceptor molecule with an acid, which are being triggered with activation of the 5' hydroxyl group. So the basic residues of the active sites of PNK, that is a polynucleotide kinase, that interact with negatively charged phosphate group of the backbone of the DNA. So therefore, the polynucleotide kinase, it actually catalyzes the transfer of the phosphate group from the gamma position of the ATP to the 5' end of either of the DNA or the RNA or the nucleoside monophosphate moiety. So therefore, the PNK can convert the 3' phosphate or the 5' hydroxyl end into the respective phosphate of hydroxyl molecule, which actually blocks the further ligation by the ligase enzyme. Now let's give you the total idea about how does this PNK basically modulate. Now, one of the most important function of the PNK is actually the one of the transfer, which is called the exchange reaction transfer, where the target DNA of the RNA having the 5' phosphate is incubated with the excess ADP, where the PNK transfers the phosphate from the nucleic acid to the ADP, forming ATP. PNK then performs a forward reaction and transfers the phosphate from ATP to the target nucleic acid, and therefore the exchange reactions take place. Now, the question lies here that whether that the two major uses of the PNK in the linker and the adapters, they are the phosphorylated alone with the fragment of the DNA before ligation, which requires a 5' phosphate group. Now, this includes the product of the polymerase chain reactions, which are generated by using a non-phosphorylated primers. So PNK is also used for radio labeling oligonucleotides generally with P32 labeled radioactive probe hybridization probe isotope. So hence you can understand that the involvement of these RDT enzyme, they play an important role in general in to bring about the labeling of the probes, the markers, etc. So it actually plays an important role in creation or conversion of a blunt end to a cohesive compatible end. Then we come to another most interesting enzyme called ribonucleases. Like that of the deoxyribonuclease, ribonucleases that catalyzes the hydrolysis of a ribonucleotide from either a single-stranded or double-stranded RNA sequence, they are called ribonucleotide. RNAs, they are being classified into two types depending on the position of the cleavage, endoribonuclease and exo, respectively. RNase is important for RNA maturation and processing. So therefore, RNase A and RNase H, they play an important role to bring about the changes. Well, to talk about the applications of the RNAs, I would like to give you a hint about it, that mainly the involvement activity of the RNAs is to use it to remove any kind of RNA contamination from the DNA sample. And henceforth, just to give you an idea about, for the construction of the cDNA library, the RNase H enzyme is used to cleave the RNA strand and basically the DNA RNA hybrid strand. So non-specific endoribonucleases that degrade the RNA by the hydrolytic cleavage of DNA RNA hybrid, an enzyme being bound to some kind of divalent metal ion as a cofactor here. So therefore, the product being formed is actually the 5' phosphorylated single-stranded DNA. So during any kind of construction of the cDNA library or any kind of cloning library purpose from the RNA sample, so therefore, when you are planning to do it, we have to use RNase H enzyme in order to cleave the RNA strand of DNA RNA duplex complexes. So therefore, we can understand the involvement of these ribonucleases in important role in DNA technology and the important role in bringing out some level of modification. So here we are here to give you an idea or glimpses, an overview about how this RDT enzyme plays an important role in bringing out in modifications, recognitions, and to some extent, modification as a whole, uh, its important role, its impact in the cloning experiments of the rDNA technology. Henceforth, I, Dr. Ray, signing out for you. Bye-bye for today. Thank you.